how cancer is studied. And some of these slides will be familiar because we've talked about different parts of this all the way through. So I won't spend as much time on each one, just kind of hitting the key points, but we'll take you through more slowly with some of the technologies that are in use, and a little bit about epidemiology, because we haven't talked about that at all, and what that is and how it connects back to basic research and translational research. And I'm hoping at the end that we have some time for hot topics, because I think there's some things as we come to the close. This was going to be our last webinar, but we've had email from people saying, as long we've got the software for a few more months, it's on a yearly basis, so why don't we add a few more webinars to the series? So we did that, and they'll be coming up. So the hot topics, if there's anything you don't see there that you think is a hot topic, and if you have any ideas on how we can maybe work with those hot topics, do we structure a new webinar around one of them? Do we write a white paper? Do we bring it back to our groups? Just some ideas of what we might do as we move forward as a group. So again, our mission is to impact all different stakeholders, and so we're conducting the webinars. The content for this section was Jane Perlmutter and myself. I put together the slide deck, and Nora does all of our editing for us. So the way we've been doing it, the last couple I think have worked, where at the end of each section we open it up for questions rather than, we can't really do it throughout, which is the way this was structured. When I do in-person trainings, it's people raising hands and we ask questions all throughout, but it doesn't really work on webinars because if the mics are open, then the interference is too great. So if you have a question, type it into the question section, and after each section, I'll look at those and unmute you, and you can ask questions. So the learning objectives for this session are to better understand the cancer research process, to better understand the importance of tissue donation, and to better understand the benefits and challenges of cancer research. So we're going to start with some background information just to set the groundwork. We'll move through basic translational and clinical research, and we're going to have a little thing on epidemiology at the end, as well as the hot topics. So Patty, welcome. I'm going to make you a panelist so that you can talk throughout if you need to. Let me go back to, once I do that, we come back out of the viewing scene. But you're open all the time, Patty.
Okay, I'll mute while I'm.
Perfect. So let's start with a little background information. I think for a lot of us, science, technology isn't a part of our real life experiences. We come from all kinds of backgrounds, but as we get into the sciences, sometimes it helps with a little bit of background. We've talked about this before. Just really quickly, BASIC is about the advancement of knowledge. It's not really there to answer a question. It's there to understand something, to find out something. People are looking at normal cells in the body, so normal prostate cells. What do they look like? How do they progress from a normal cell to a cancer cell? So in the BASIC area, people will be looking at that. They're wondering how things work. What's DNA? How do those mutations work? How do the pathways work? How are the genes involved? What's happening with the proteins? So they're just looking to gain basic knowledge. And sometimes something pops up and they go, whoa, that may play a role. And so they move it further and they take that idea and they bring it into the translational realm. And as that idea gets built on and more knowledge is discovered around it, they can actually start testing the idea in lab animals, and it's usually mice. If it passes that mouse test, if it gets through the translational area, then it moves into the clinic. And that really is, where we become involved as NCTG advocates. A lot of us are involved in other stages as well with other hats on, but we're involved as it gets into the clinical trial protocol development stage. Epidemiology is sort of after the fact, where researchers gather information from large groups and they start asking questions, and it's sometimes about cause, sometimes about why, sometimes about how, and we'll go through that a little bit more. But as epidemiologists find out relationships and connections, then they feed those questions back to the basic researchers and the translational researchers and back into the clinic again. So epidemiology is sort of a place where questions are generated and fed back through. And that's the pathway. We should have an arrow going from epidemiology right back here again, because whatever questions are asked here or formulated, then they're actually tested back in the basic translational to clinic. And again, I just like the images of knowing we're working at the bench, we're working in animals, moves to people in clinical trials, and finally to the bench, or finally to the bedside and the general population. So what is the scientific method? We talked about this a little bit before, so this is a review for some of you. But essentially, it's followed in every single one of those little arrows, whether it's a basic, translational, clinical, or epidemiologist. It's a method for finding an answer. And you begin by observation. And you begin to document what you know. Then you hypothesize and you ask a question. Then you test it. You collect the data, analyze it. Then you form a conclusion. And so this happens all the way through, including the clinical trial stage. So let's move on to basic research and talk a little bit about what that is. We've shown this slide before on how tough this really is, that basic science really needs to continue to be funded, because here's where everything starts and moves forward, but it's very incremental. It's not there to give you an answer. It's there to form a platform to ask questions. In vitro means research in test tubes. So people are right at the bench in the lab, and hopefully they find something that moves forward into the clinic. And as we progress, more and more disciplines are coming into cancer research. So we have physicists now. We have cell biologists and chemists, immunologists, molecular biologists, bioengineers who are putting together these mouse models, bioinformatics people who are crunching the data, systems biology people who are taking it even wider, and we'll talk a little bit about that. So more and more disciplines are coming into this field. In fact, I was just involved with a project where there was a math modeler who was putting together, and she again was crunching data, but making mathematical models that would help determine risk. So lots of different people involved with cancer research than 20 years ago. We mentioned systems biology, and this is still relatively new. And if you can look on this image, the life sciences, 15, 20 years ago, it was all here. It was genetic modification, measurements, hypothesis. All of the basic research was done there. And then as more and more knowledge became available, they brought in other people. They brought in informatics scientists who do modeling and have databases, and then the system scientists who also do some analysis and modeling and synthesis of all the data. So kind of a new approach, and the NCI is continually broadening the spectrum of the kind of people that they pull into the realm to really help this move quickly. Rather than looking at one thing at a time and then another thing at a time, taking 3 years to do that, and then moving to the second one for 2 or 3 years. They bring in everybody all at once. And that takes a little education for an engineer to understand what an immunologist is talking about, and an immunologist to understand what do I care about bioinformatics. So there is a period of time, and we're a little bit in that time where everybody's learning what everybody else does and how they can help them. And the purpose of that is to get it done quicker. Let's move through this question research phase as quick as we can. So let's talk a little bit about the tools of the trade, and that's cell lines, and we'll talk a little bit more about that. And they can be either animal. In fact, 20-some years ago, it was all animal. But we've now got human cell lines. They're kept alive in plastic containers. The technology is moving forward in all kinds of ways. But like Patty mentioned, I think it was the last one, pathology will always be involved. You always have to have a pathologist look at the tumor as it comes out of somebody. We have a lot of pathologists who are now researchers because they understand at that cellular level of looking at under the microscope a lot more. So now there's pathologists who aren't there at the clinical end, but are actually at the research end. And so it's a merging of a lot of different people. So let's talk a little bit more about cell lines. These are just a few. MCF7 is the big breast cancer one. There's a couple prostate ones listed here, and acute myeloid leukemia. There are other ones, just because of space on a slide. I picked a few. When you're involved with the protocol, and if they're looking, this probably won't be at the clinical trial area. This will be more in the basic and translational area. But if you're involved at that level, or if they mention it in the protocol that their background, it would be in the background information section, that they have data from cell lines, what you would kind of want to know is, are those lines human cell lines or animal cell lines? So you would hope that they would be human, because then the correlation is right there. Animal, you have to make inferences. Are they cancer cells or normal cell lines? It would be nice to have both. More and more that's happening, but it's been really tough to get normal cell lines. And how many cell lines are they using? The more the better. So not only do you have these cell lines, but somehow you have to keep them in culture. And how are the cells grown? We started out with 2D, and you can see from this little image that it's the easiest, it's the least expensive, but it's not very much like the natural environment. It's in a petri dish, it's in a plate. Then again, you're making inferences. We moved to 3D, and that is better, but it's more expensive. It takes more technique. It is more realistic because you've got a 3D model of what's happening in the body, but live tissue is always the best. And that's why the tissue donation becomes such a big part of any of these talks, because if you can look at something, find out something, test something in actual human cancer tissue or normal tissue, it's much more illustrative of what's really happening. So let's talk about some of the tests. And again, when you're reading a protocol, in the background information, they may mention this, that immunohistochemistry, and the acronym is IHC, and oftentimes all you see is the acronym. They are assuming you know what that means. But what it is a lab test that stains tissue from a biopsy, and they're using a stained antibody to detect a specific protein, which is the antigen. And you can see, we've talked before in immunology, where the antibody will attach to the antigen, and then if it's got a fluorescent stain tag on it, you can see it under a microscope. And so they use this in HER2 and a lot of other places. It's a test that's quite oftenly used. And here's a nice image of these pink ones would be the HER2 cells that are there. And there would be HER2 positive here. So there's a lot of different areas where they use immunohistochemistry. Another area that is used a lot and that we actually hear a lot about is DNA sequencing. And really all that is a method, it's a lab test for determining the order of nucleotide bases, the adenine, guanine, cytosine, and thymine. How do they line up? And here's this little picture. This is the printout that you get. And from the DNA sequencing, you can hopefully improve the diagnosis. You can get a better diagnosis of disease. Hopefully earlier detection of genetic predispositions to disease. You might be able to assess risk for certain diseases, and this would be a risk-based approach. It helps with rational drug design. If there are specific genes that are causing disease, and there are some people who are working at replacing defective genes for certain diseases. And we've talked a little bit about pharmacogenomics, customizing drugs, and it's based on the DNA sequencing. So they take someone's biopsy and they sequence it and find out what's happening there. And, when this first started, it took a long time to sequence anybody's genome, and it was very expensive. And now we're down to really quickly it can be done, and it can be done for an amount of money that makes it plausible. So it's used not only in research, but now it's found its way into the clinic. The tough part, the part where we are now, because the technology is there, it will continue to get a little bit cheaper, it'll continue to get a little bit faster, but where we are now is, what do we do with all of that, Dave? What do we do with all of these sequences? How do we interpret that? How do we really find out what's clinically important for somebody? Where are the changes that will affect an outcome for somebody? So this is where all these math modelers and bioinformatics people are working, taking all these realms and realms of data and trying to crunch through that and bring ideas back to the clinic of, here's where it matters. If you see this sequence, here's what happens. So that's where we are with DNA sequencing. Another term you may have used or heard is fluorescence in situ hybridization, or FISH. And again, it's a technique used to detect and localize the presence of specific DNA sequences. So immunohistochemistry, it's a fluorescent dye, and you're looking at protein. In FISH, you're looking at genes. So it's a little bit more specific. It's a little bit more on target. Here you can see, so the chromosomes are in blue, and the red and green spots are the mutated genes. And again, that's used in HER2 and other places. So it's another test. It's used both in research, and again, it has moved into the clinic. People will see this on pathology reports, and treatment will be based on what these tests are showing. So as you review protocols, if you see this in the background information, this is what's happening, and this is why they're doing it. Microarrays, we see a lot of information on microarrays. People will talk about that all of the time. It's quite a bit more complicated, too complicated to go through a whole lot here, but essentially you've got normal tissue and you've got tumor tissue. You put them in these little wells. You put a dye on there again. They get on these little chips, and these chips are now read by a machine. And instead of looking at one gene like HER2, you're looking at maybe 500 or 1,000 all at the same time. So it's much more efficient. Um, they've got little chips for different diseases, and so here this is separating out what kind of lymphoma someone has, and it's all based on the green genes show up, they're underexpressed. The red genes are overexpressed. Whatever pattern shows up determines which type of lymphoma you have. So it has moved, again, out of research into the clinic. It's still used in research to move us there. This is where they still get a lot of information from. But eventually it does move into the clinic. And it's a wonderful way to help a doctor know what they're dealing with. So that's a little bit on microarrays. All of this, in fact, all of the ones, all of the technologies we've talked about, if you go back to our website, we have maybe 5 pages on this. So it goes in much more detail, lots more pictures, lots more explanation of what's happening, why they're doing it, what's the problems with it, what are the advantages of it. So if you like this kind of information, just go back to our website, and there's lots and lots of information on it. So here's kind of a little summary of just a few of the research tools that are available. Some are looking at DNA, some are looking at RNA, and some are looking at proteins. And we have a lot more on our website with all of these. So as an advocate, our primary focus and goal and role is to represent the patient. And if you're involved with basic research, here's the kind of ways you can be involved. You can be a legislative advocate for basic research. You can help prioritize which ideas move forward. You can be on grant reviews. You can be a consultant on a graph. You might be a member of a SPORE or some other research program at your cancer center, or the NCI, or some other advisory committee. So in the realm of basic research, here's where advocates kind of can play a role. So we're at the end of this section. I don't see any questions, so I'll just keep moving forward. If you have a question, though, if you think of one, just type it in and we'll go back and get it. So let's move over to translational research. And again, that's where the basic ideas come into animals to get tested. That very first testing that's done to see if the hypothesis that was generated here holds up in animals. Some of you may have heard of the NCI roadmap. It started around 2003, 2004. It was a 10-year project, ended in 2014. And it was really a new model to move from individual researchers sitting at their bench doing their own experiment to team science. How do you bring more people involved? How do you bring more people involved with the idea that your idea is going to move into the clinic? There are some people who can spend their whole life at the bench, looking at tumors and just wondering, what are they? Why are they there? And they never get past wondering about telomeres, or it could be anything. I come from the research world. You can take any one of us and put us down with a microscope, and we'll sit there, and whether it's counting how many legs are on a caterpillar, people get excited about it, and they can spend their entire career just looking at that. And the NCI goes, no, this isn't going to work anymore. We're not getting better outcomes for people. So they brought basic researchers and formed teams of people who wanted to move those ideas. So there was early translation, new partnerships were formed, there were phase 1 and 2 trials to test ideas quickly, so that some of you may have heard of Liz Blackburn. She is prize winner. She's a researcher at UCSF, and I was at a talk she gave one time. And the purpose of the talk is not only to present your ideas, but also to get feedback from people so that you can be improved. There's a whole lot of that that goes on in the research world. And somebody at the end said, you know, you went from A to C, and I didn't see any B in there. How did you make that leap, and why did you make that leap? And she said, you know, I could have a grad student do that. I don't need to do that. I know what's going to happen. I knew we were going to get to C and then D. So I'm skipping those 4 years of looking at B and letting a grad student take that as their project and idea. I'm going to move forward and get to an answer, a clinical answer. And that's part of what this translational continuum is about. It's to move some of those basic ideas a little bit more quickly to the clinic and let somebody else keep studying, keep looking at, keep wondering about the basic part of it, but move it through. Get it to a point where it's going to make a difference for. I went back to the NCI just a couple of days ago when I was getting ready for this talk to look at this to see if I could find any information about it, and that's gone from their website. I mean, after 10 years, they take it down, they've moved on to something else, and you know, I couldn't find anything else but some papers that were written about it. So when we get to the end, if anybody knows anything about, you know, the results that came from here, other than the fact that it was a process that did work, we have integrated this into the research continuum. The networks are part of that, so that the networks have now been redesigned to get some of these basic researchers and the industry involved with the questions that are being asked in the network, not just from the clinician anymore. Some of these are coming from other places, from SPORES and from other places. So there was, even the network had an outcome from this roadmap. So again, the reality is that there are more failures than successes. It's just time-consuming, tough, really hard to move ideas to a place where it has an impact. And sometimes it's the new intervention is too toxic, or maybe it's not effective. So all of those great ideas, we've talked before about that funnel, how you start out with thousands and end up with tens. It's a long, hard, tough process. So let's talk a little bit about the animal models that are used. You'll see the term in vivo, and that means research in living organisms. That's what in vivo means. In vitro means the research is happening in a test tube or a glass petri dish, but vivo is a living organism, and it's usually mice. And that's just because they're cheap, they've got a short lifespan, so you can get the answer really quickly. There's lots of different types of mouse models. We've got genetically altered mice, specifically around a certain question that's being asked. You can have an immune deficient mouse. What happens if they have no immune system left? Xenografts, this is a picture of a xenograft where a human tumor is actually implanted in the mouse, and then the intervention can be tested. So the drug can be administered to see if it has any effect. So mice are usually the animal of choice. And again, back to tissue research, that the more real human tissue that we have, the better that is. We can put the human tissue into that mouse and see what happens, but then you're making inferences again. So it's important to have human tissue. We're going to get biomarkers there, hopefully find out who will experience drug side effects. The tough part is that we don't have enough tissue donated, and the processes involved haven't been totally standardized, from collection to processing to analysis. And all kinds of people are still working on that, but every time you see a protocol where they talk about gathering samples, biopsies, um, make sure that it's done in a central lab. If there's 20 sites or 100 sites or 500 sites, the first question you ask is, do you have a central lab that's testing them? Is it a CLIA licensed lab? When this correlative science was first started, even 10, 12, 15 years ago, every site was doing it themselves, and they would get together and they would try to merge the results, and everybody did it a little bit different, and it was apples and oranges, and it was all wasted time and effort and tissue from people, because unless it's all being tested exactly the same, under the same rigorous protocol, you just can't draw conclusions. So as an advocate, the first question you ask is, do you have a central lab? Here's an example of, and this actually came from a cooperative group clinical trial, uh, a correlative science add-on to some clinical trial that they were doing. And this shows that for colon cancer patients who have KRAS mutations, their survival is not as good. And it not only showed that, but they also found that people with KRAS mutations had no effect whatsoever from the two drugs that were used at the time. So before this was known, before we knew about KRAS mutations, people were getting these drugs. And if they had a mutated KRAS, they had all the side effects and none of the benefits. So today, they'll be tested. Their tissue will be tested. If they have a KRAS mutation, they won't receive these two drugs. In the meantime, pharmaceuticals industry came up with new drugs to treat people with a mutated KRAS. So that's why it's so important to do the correlative science. That's why it's so important to get donated tissue in a clinical trial, because we find out a whole lot from that. And you never know what it's going to be until you start the test. There's all kinds of hypotheses. They know when they write it up, here's what we're hoping to see. And sometimes they see that, and sometimes they see something else. So this is just one example. There are lots of other ones why it's so important to get that tissue donated. Again, this is a slide we've shown, and it's, we're kind of here. We're not really here. We're more kind of in the middle now. We've moved for most patients. Well, no, we probably should say for some patients. Some patients are still treated this way, where each tumor gets the same standard therapy. We don't have any knowledge of the subgroups or anything else. And they reported out in 2001 that only 1 in 3 patients benefited from cancer drug therapy, from chemo, because they were getting the wrong drug. They were getting a drug that did not work because of their particular tumor. The future will be, each one of these will have a molecular diagnosis. They may have other tests run on them, whether it's DNA sequencing, whether it's FISH. Something will happen in here such that they will be identified for which therapy will work for them. And that's the goal of personalized medicine. That's where we're moving. And that's why, again, why tissue is so important. So let's talk just a little bit about how can an advocate help at the translational level. And you'll see that these are exactly the same as at the basic level. If you go up to the Hill and do legislative advocacy, and Jack Whelan is going to talk to us about that on our next one, our next webinar, he will be leading, talking about that. And the reason why is because as funds go down, depending on who's in Congress, depending on who's the president, there are people who believe that research should happen at an industry, that it shouldn't be publicly funded. And we'll talk a little bit more later, but there are things that happen with publicly funded research that will not ever happen with industry-funded research. So it's very important for us as advocates to continue to um, let our senator or congresswoman man know, and our president, when we're voting for president, ask them these questions. Do you support government-funded research, NIH, NCI? It's something that most of us don't think about, but it absolutely pays off in the end on how many dollars are involved. Again, we can help prioritize which ideas move forward. I think this is one of the most important roles that we can play at the basic and translational level, because um, researchers tend to get excited about their area, and again, they forget a little bit about the patient and the outcome. Is this really going to make a difference for some patient? If it's not going to do it, let's keep it in the basic realm. Let's somebody else look at it, but let's only move ideas forward that's going to make a difference for somebody. So same with grant review. We're in a way we're prioritizing which gets funded here as well. Here you do it within committees with SPORES and whatever committee you're working on. Here you can actually do it with your vote. We're beginning to work as consultants on grants, and then we can help that researcher all the way along, letting them know, okay, if you've got two questions, this is the question that matters to people. This one, not so much. If you've only got limited time and money, answer this question. So we can really help through basic and translational research in these areas, again, as members of SPORES or on the NCI. So let's see, we've got a question from Jack, so we're at the end of this section. So Jack, let me, let's see. I'm going to unmute you, Jack, so you can ask your question.
Well, it wasn't a question, Peggy, although I'm really appreciating the presentation. It was just a hint that someone once passed on to me when you had mentioned in vivo and in vitro, and the hint was that someone told me to think of in vivo as in Vivian, so that you'd kind of remember it as something being studied in living versus in vitro. And so I just passed that along.
Perfect. Nice tips. It's always, when you're coming into something and you see these terms, you've got to say something in your head that's going to tip that into a meaningful, what does that mean? So that's a great tip. So anybody, as we move forward, this is the kind of platform where you can offer those kind of tips, because as advocates, we're here to help each other. So if you've got a tip on something that's for anything that we're talking about, let us know. So Jack, I'm going to mute you again, and I'm going to unmute Dorothy. And Dorothy, why don't you go ahead?
Yes, one of the things that occurs to me is that in the past we didn't have means of separating mutated versus non-mutated, and there could very well have been drugs that got dropped or weren't approved because they didn't appear to have a significant effect. But if you then went back and sorted those out based on what we now understand about whether it's DNA or other aspects, is there any effort done, whether it's an industry or otherwise, to go back and look at some of those compounds now that they can be distinguished better?
Yes, in fact, both the NCI, cancer centers, and industry are madly doing that, and there's databases that have all of these drugs, and there's databases where they're feeding in information like crazy about which pathways that each drug affects or which mutation, and there's people doing that. And for some, they can just bring that drug forward without any other further testing. They now just have it and put it in a clinical trial to confirm that. For some, they get ideas. They go, whoa, this is close to this, and there's that mutation, and it goes back into a translational aspect where there's more research, and they tweak it a little bit to do that. So both things are happening, but for sure we are getting more and more drugs that had been phased out, that are now back, and most research dollars have already been spent, so this is a really cheap way to get some new good treatments out there.
Yeah.
So yes, that is happening.
Okay, great, thanks.
Okay, let's just keep moving forward. So now we're going to talk about clinical research, and this is where we're most involved as network advocates. This is why we're taking these websites or webinars. And this, again, this is a slide that I've used before, but it's so important for actually the general public to understand that we don't move forward until we get clinical trials. And just a successful mouse experiment isn't going to mean that it's going to be successful. It's still in that funnel, and more ideas will drop out than reach the end. So I use this slide every time I'm talking to the general public or to anybody else who wonders why this is so slow, why they just read about some wonderful new drug, and then I'll ask, did you happen to see whether that was in a mouse model? And usually they don't know because they haven't read that deeply or they don't remember it. But I let them know that that's where a lot of the reports that we find in the paper are from. So feel free to use any of these images. So again, the reality is that it just takes time and it takes money, and most of these just don't end up being successful. And even if it is successful, then it takes a whole lot of time to be approved by the FDA. And then it takes a whole lot of time from the time it's an FDA before it gets out into standard practice. And that's why it's so important that we have second opinions, that researchers know the newest and the latest, but the community docs don't sometimes know it for a while. They have to read it in the literature, and that takes a publication, and that takes another year or so. And so this whole process is just really long and slow. Again, there's most people, when they think of a clinical trial, they first thing they think about is a new drug. And there definitely are new drugs being tested, but there are also prevention trials. There's screening and early detection trials. There's detecting better diagnostic trials. And of course, there are treatment trials. And some of them are for new drugs. Some of them are for targeted therapies. Some of them are surgical procedures, a sentinel node versus complete node dissection, partial mastectomy versus full radical mastectomy. All of these came through clinical trials. The part, and the same thing with radiation, all of them, whether we had the full chest to a localized area, how much, protection things, all of these advances come through a clinical trial. What Patty has helped me understand better and provided a lot of good information on is the importance of publicly funded clinical trials. And that's because comparison of existing drugs, and Dorothy, some of this is your question. They're not the old ones that are being brought forward again, but maybe there's 4 drugs out there, and which one? And they're all treating the same thing. Which one of them is really better? They're all from different industries. Industry is never going to compare those, but the NCI will. So it's important to put those through a clinical trial, and that's really tough to do. Only government will do that. Industry is not going to do it. The other thing they're not going to do is which order is better. Do you give drug A followed by drug B, or do you start with drug B and move to drug A? Or do you do them in combination? What is the best dose? More, less, longer, smaller? All of those are kind of questions that the government will ask, NCI will ask, public funding will ask. Industry won't ask that. They won't ask those kind of questions. So it's another place, if you speak with community groups or the organization you're from, help them understand how important that publicly funded research is. And again, quality of life and survivorship is another kind of clinical trial. Sometimes it's an add-on. More and more, whether it's patient-reported outcomes, really important information. Because as there's 3 and 4 drugs available, and maybe survival is the same for all 3 of them, but maybe one of them has a much higher quality of life than the other 2. And you're not going to know that until you study it. And patient-reported outcomes is one of those. I was at a meeting once. We were in Florida. It was hot. We got out, and everybody wanted to go into the pool. And one of the guys that was in the meeting with us, he said, no. Everybody kept asking him why. He was on a drug that gave him a really bad rash. Now, his doctor didn't, he said, it wasn't a grade 4. Maybe it wasn't even a grade 3. So it medically didn't influence the doctor. He had no concern about it whatsoever. But for this guy, this man, he was not going to work, and he would not go swimming because the rash he was so embarrassed about that. So he wasn't even working. He came to the meeting. He was perfectly healthy, but the rash bothered him personally so much that he wouldn't do something. So doctors have to know that. Doctors have to know the kinds of things that matter to patients. And that's one of the things we can keep reminding them of with these quality of life and patient-reported outcomes. It matters to people what happens to them, and it's individual. We can't generalize. And the only way to get that information is back from the patients. So that's why these are so important. So again, just a little summary. We've had a whole entire talk Patty gave on clinical trials, so we're not going to spend hardly any time at all on that. But the take-home for the phases is phase 0, does it hit the target? Why spend time and money if it doesn't hit the target? So just get that question asked and answered really quick. Phase 1, find a safe dose. Phase 2, does it work? Phase 3, is it better than standard of care or usual care? And phase 4, are there some subgroups of people out there where we're starting to find other either side effects or successes or failures? So we keep finding data. That's it in a nutshell. That's kind of what's happening. And this puts it into a little bit wider frame where you've got the number of people, the length of time, again, why it's being done. The success rate, you can see phase 2 and phase 3, it's really tough. Look at how many drugs have to go through there before we get a successful answer. And phase 4 really is just gathering more information. So the barriers to accrual, I didn't put it on here, but I think one of the biggest barriers is the study design. Is it going to matter to patients? If it's a well-designed study, usually it accrues. If it's not, it's a whole lot harder. But there are other barriers. Physicians sometimes won't talk about them. Patients, if no one suggested it, they're not going to know about it unless they're the unusual patient who researchers options before they start treatment. And for a lot of patients, they don't start doing research until after. For some people, it's just too many decisions to think through, and there may be cost or time constraints. One of the big areas for our nonprofits is health literacy, because I think if the general public understood what a clinical trial was for, and that all the drugs that they're benefiting from came through a clinical trial, not some of the older ones. Aspirin was never put through a clinical trial, but anything that has been discovered in the last 40 years or so, maybe a little bit longer from that, has come through a clinical trial. So they're benefiting from somebody else having volunteered. And we just don't have that in context as part of our basic understanding as community members. We don't know the role of clinical trials. And I think it's way too late once somebody's been diagnosed to learn about a clinical trial. Then they're trying to understand, what does the randomization mean? What does genomic targeted therapy mean? The health literacy aspect, I think, is critical for all of us. So anybody that's on the call, anybody who hears this webinar afterwards, please take these slides. You can get copies of them. You can send the webinar URL to anybody, and help your community and organization understand the importance of cancer research funding, and clinical trials and tissue donation. I think as advocates, we need to get a little bit wider and help the health literacy problem in our country. Benefits and risks, there are some benefits. Usually they're for future patients, although some of the targeted therapies, there will absolutely be benefits for those specific people. You can also start talking about the benefits of donating tissue. Now, it may not, again, be for you individually. It may not be for that person that enters that clinical trial, but it will definitely help people in the future. So that's a benefit that matters. There's been some recent literature out that people who have enrolled in clinical trials, that was one of the primary reasons, because they wanted to make a difference for somebody, for their kids, their grandkids, the community in general. So it's important. We almost always just have one sentence in an informed consent document. It may help future patients. I think we can write a paragraph or two about that. I think we can help them understand what personalized medicine is, precision medicine, and how the tissue is important, and how the clinical trial is important. I know there's all kinds of legal issues that have to be considered, but I think we can move from one sentence to maybe a paragraph about some of these future benefits for patients. For risks that people absolutely have to understand, that there are some risks involved. Usually it's just that it's not as good as usual of care, but if they move on to standard of care, that's fine. There may be some unknown side effects, and there may be extra cost visits. For some people, the extra office visits they almost like, because they know that a doctor's looking at them. They're looking at them much more closely than getting chemo and 3 weeks later coming back in. So for some patients, I've talked to people who say, no, I actually like having to come in more. So, it's different for everybody. I think ethical issues are really important. Again, for patients, they want a better outcome. That's what they're hoping for. So we have to understand and help them understand that they may have a better outcome, but they may not. There will be some loss of privacy because data is going to be shared. It may cost more, and there may be some genomic privacy loss. And there's all kind of built-in things so that these don't go outside the research or clinical area. So usually people understand, well, we have to send it to this committee or that committee or this committee. But there are some people who don't want this in their medical record. So you can see the list is much, much longer here as clinical trial advocacy. We not only have all the ones we talked about before, but now we can be members of IRB, Data Safety Monitoring Board. We can be on FDA committees. We can assist in the trial priority, what's moving forward in our committee, whether that's through the concept level or the protocol level. We can assist in developing the protocol to make sure that any barrier that's there, whether it's extra visits, whether it's extra imaging, whether it's sometimes they ask for 5 biopsies and maybe really they only need 2. Sure, they would love to have 5, but if that's prohibitive for a patient, maybe 2 is enough. Maybe you don't really need all of those other ones. So all of that stuff, all the requirements, those are areas where we can have an input and we can help them get it over a barrier. We can help them write the consent form. And Dorothy, here's a place that sometimes advocates get left off these committees and they don't see the consent form till it's all done, and that's just a shame. And sometimes that's because the consent form is written really quickly and it's a cut and a paste and it looks like every other consent form, and so that's where you can have some real discussions with your team and say, you know, we want to get that standard form that you've been using and we want to fix it as much as we can, and then we want to look at each one, and so if you've got breast advocates and lung advocates and GI advocates, whatever comes through that group, see if they can't get the consent forms beforehand, and that includes the tissue donation forms, so that it's clear. It can be read by a lay person and understood, and that there are examples of what's happening in a way that's not technical in nature. So kind of summary paragraphs saying, here's what this last 3 pages was really about. And sometimes they'll let you do that and sometimes they won't. And sometimes you'll get it done and it'll go to a site and the site will totally change it. Because each site essentially has their IRB, and some sites are very strict about how that looks. And so all we can do is our best, but I think we can at least say, can we take a look at that and see if we can clean it up a little bit. We can also assist in developing the education materials, and that's to be on the committee that's writing them. That's not having them sent to you after the fact for you to proof. Because a whole lot of times when you get them, they're not willing to make the change. Either it's too far down the process, they run out of time, there's not enough space to change this word to that word or whatever word. So you want to be on the committee that's developing, not the committee that's proofing them. And we can also assist in disseminating results. And we can make sure that on our group website that results are publicized, that there's a lay summary if possible. The Alliance has that in place where lay summaries are written for not every study, although we hope to get there, but the big, large ones where lots of sites are involved. We have lay summaries written. So we can ask to see if that can't be put on websites, your group's website. So you can see there's a whole lot more going on here. Let's see, we've got Dorothy. The challenge of more visit impacts potential to recruit from more rural, underserved areas and also from less affluent who can't afford to take time off. Yes, it's absolutely true. And there's been all kinds of studies where women will choose a mastectomy over lumpectomy and radiation because they can't go in. They just can't do that. So that's absolutely true, that those extra visits will drop off people. I've moved 10 years ago from the Bay Area to a rural community, and I now know what that is like. It absolutely does impact what's going on. So thanks for that reminder, Dorothy. It is true. There will be people who will be lost because of those extra visits. Dorothy said she absolutely agrees on the consent forms. So Dorothy, I'm going to unmute you and you go ahead and just say what you want to say.
Well, where I've been involved in advocacy through the particular group, I haven't been involved, and to my knowledge, none of the other advocates have been involved. And I think that we really have a very significant potential role in explaining things in a way that makes sense to a patient. And that's, I think, the earlier the involvement there, the better off we are, and the better off the trial crew is, because
Yeah.
it can only help with that. So that was all.
Okay, good, thank you. Okay, so we're going to mute you again, and we're going to move on. And this might be new for some people, epidemiology research. And I think this cartoon, just every time I see it, I have to laugh, because the people at Pratovilla have low rates of cancer. It must be all the sarsaparilla they drink, unless it's all the pickles that they eat. Unless it's the mayonnaise they put on the pickles, unless it's the chocolate they dip the pickles in, and on and on and on and on. So epidemiology puts two ideas and they draw inferences, and sometimes those inferences aren't really correct because they just don't have enough data. So we'll go through this a little bit more so you can see. Here's what an epidemiologist is attempting to determine. It's a who, what, where, when, how, and why. Who's prone to developing cancer? What exposure do cancer patients have in common? Where is the risk the highest? When is cancer most likely to occur, and what are its trends over time? And how and why is the risk increased through exposure? So the most successful epidemiology outcome has been lung cancer was caused by smoking for most patients. There are absolutely lung cancer patients who never smoked and weren't exposed to secondhand enough to get the lung cancer. It's a different form, and they haven't figured that part out yet. But that was the first time that an epidemiology study pointed, and so they gathered the data, they found it, they sent it back to labs, and because it was so politically a hot potato, it took years and years and years. It's kind of like climate control now. How many dollars can be spent disproving something that somebody else has proved? And so we're stuck in that there, and that happened with smoking. But that connection was made because of epidemiologists who went through the records and found a pattern with people who smoked and then came down with lung cancer. And so that's what this is. We're looking for a cause, and it's very difficult to accomplish. The reason that the smoking one was so successful was because there is a direct correlation, and for a lot of things there isn't a direct correlation. There's usually no randomized trials. There are always lots of variables that aren't accounted for, and you need lots and lots and lots of numbers to make these correlations hold up. So here's the kind of studies, epidemiology studies. It turns out that 99% of them are these observational analytical studies. And in observational studies, there can be a comparison group, and it can be a cohort, a case control, or a cross-sectional. And this, so a cohort is looking at exposure and outcome. Case control looks at outcome and then exposure. So here you're looking at if they've been exposed, what happens. In a case control, you go back to the data and you say, here's what happened. I wonder what they were exposed to. And then cross-sectional, you're asking both questions at the same time. It's real tough. There are very few experimental studies because then you're randomizing, and you don't want to give somebody an exposure to something. You don't want to have a group and say, okay, now 100 of you are going to smoke this many cigarettes a day, or 100 of you are going to be exposed to benzene. So we don't have very many studies like that. Almost all of them are analytical and observational. And that, so 99% of them are. And again, the cohort is did two groups, and one is exposed to something and one was not exposed to it, and did it matter? In the case control, it's why me? There's two groups, one with the disease and one is healthy, and you're asking why. So let's go back here a little bit. Whoops, we went the wrong way here. So let's get back to here. In fact, let's go back to here, because the concern for us as advocates, I think, is a lot of the data that comes out, whether it's obesity and exercise, a third of cancers can be prevented. That's some of the epidemiology data that has been coming out over probably the last 5 to 10 years. And I looked really hard, and I talked to a lot of people, and most of them said of that 1/3 that can be prevented because of lifestyle, most of that is stop smoking and don't be exposed to infectious agents, whether it's PVC or whether it's any of the infectious agents, hepatitis C, that people will get cancer from. So those are two that are direct correlations, and they know if you stop doing those. But all of these other ones, whether it's alcohol or calories or just recently we had one on processed meats and meats in general, and all of those numbers are down. So the processed meats was like 18%. If you stop eating this, your colon cancer risk will drop 18%. So I listened to every story, I read every article, I really looked at it, and not one of them made the correlation. So that's they were giving you a relative risk number, 18%. Nobody said what your risk for colon cancer was or is. And so I went to CDC, and it took me a long time to figure all this out. And I never really figured it out because all this charts here, there, and everywhere. But it's about 10% over your lifetime. You have about a 10% chance of getting colon cancer. And usually there's some mutation involved with that. But if 18% risk and you only have 10% chance, then you've got about, if you eat processed meats, maybe 2% more. So you go from 10% to 12%. But that's a 2% difference, not an 18% difference. And so it's really frustrating, I think, for the general public to realize that when people are talking these numbers, it's relative numbers, and they never talk about what your risk is beforehand. I was on an Institute of Medicine study looking at environmental causes of breast cancer, and I think it's 12% increased risk if you drink alcohol, somewhere between 12 and 20. That number is always changing. But again, your risk of getting breast cancer is 10. So 10% of 10 is one more, so it's now 11%. But 1 is very different than 12. So we never get told this when we read these articles, and it's one of those areas that I kind of get all hot about, that I wish that medical writers did a much better job helping people understand what this number really means. The other tough part that happens with epidemiology studies is that they're observational in nature. And what that means is somebody's going through medical records, and they're looking at medical records, and they're gathering the data to see what somebody either did and what the outcome was, or what their outcome was and then what they did. And so it's based on questionnaires. It's based on what people can recall and tell their doctor. All of that, is very iffy. So epidemiology studies are the lowest level of proof. And it's because there's so many biases involved. You're asking somebody to remember and to be honest when they talk to their doctor. Some of those happen, sometimes they don't. The other thing is there's all kind of variables that don't get looked at. What subgroup of people would have changed that? What mutation may have played a role? And so here's my hope for epidemiology. And most of the people on that IOM study were epidemiologists, and they're all excited that as biomarkers become more frequently used, and as that gets into the medical records, epidemiologists can pull people with certain biomarkers. They can identify these subgroups of people and then make those correlations much tighter, much more meaningful if they've been exposed to a risk. So that's very exciting. That's going to take some time to get, far enough down the years that all of this data gets into people's record. But then it'll be a biomarker, not somebody's recollection. Also, there's now new technical machines. Rather than asking, have you been exposed to benzene? Some people remember, some don't, some never knew. Now there will be a test. You can measure from their blood or somehow whether they've ever been exposed to that. It'll pick it up. And so some of these exposures will be absolutely tied down to a number. Your lifetime level of fat will be tied down to a number from a machine, not based on calories or what you've been eating. If you ask somebody, do you eat hamburgers every day or once a week, that answer is going to vary depending on the person and how honest they are. So I'm really excited that these two are going to be in play, and epidemiologists will have much, much better data to form their inferences from, because it's an important area of research. They're the people taking large groups of people and looking at these questions. And as they get the data, then they feed it back to the basic and translational researchers. So as they tighten up their information, better questions will be asked and answered. So I think this is very exciting for epidemiologists. So in summary, cancer research is, we know it's critical. We know it moves through a continuum. We know it requires tissue donation, and that tissue has to be standardized, and that it takes a lot of money and a long, long time, and the support of a lot of people. So that's pretty much today's talk. Here's the hot topics. So if you have a question, why don't you, what I'd kind of like to do is just open this up for anybody to ask a question. How do we ensure that the focus is on trials that change clinical practice? As an advocate, what can we do here? What can we do within our group? Do we do that through our vote on the protocol? Is there anything else we can do? How do we encourage collaboration between network groups? It's silly to ask the same question in 3 different groups, because we're just cutting the people available down to a part where accrual will really slow down. So how do we collaborate across and between the group and industry in a way that really important questions are still asked, not just does my drug work? What do people think about mandatory tissue donation? That's been a hot topic now for quite a while. And every year we get farther down into precision medicine, we see how important it is to have tissue. But we're asking a lot from people. So how do we talk about this? When is it mandatory? How often should a biopsy be done? How do we increase health literacy? This is my favorite buzz one. This is the one our nonprofits really, really, really focused on. How do we help the lay general public understand clinical trials and cancer research? What can we do around accrual issues? Whether that's within committees, within the advocate group as a whole. um, health disparities issues. How do we work on that within our committees? Do we talk about high cost of drugs? Jack is going to talk about legislative advocacy and the role that plays. And Carol wanted people to know that there are new funding streams out there. So Patty, when you look through that list, do you see anything that we should do another webinar on, or what are your thoughts around this?
I don't know. I mean, I think one of the things is trying to figure out how we're going to operate within this new NCTN system as a group. And one of the things that's been initiated is an accrual core team. So maybe we could have something around that type of thing for NCTN advocates. But I'm not, that's such a broad issue too. So I'd like to hear what others think that we can do to kind of bring everyone together and work on some of these key issues and having more of a say within our groups, maybe sharing best practices.
So if someone wants to type something in, I love the idea of best practices. I love the idea of, and I also love the idea about the accrual, because as your committee moves forward, the best thing is to get it out as wide and as deep as you can, as fast as you can. So we can do that. So we can put stuff on our website. We could put a section on our website called best practices. We could put something on our website called accrual tips. If people aren't quite thinking through it now, why don't you email Patty and myself? Would you be okay with having CISN put something on their website about this? Is there a way, maybe if we have a Dropbox for network advocates, maybe we start a Dropbox folder. I don't know, Patty, because there's people who won't want it on our website, and so
right.
I want to do it in a way that it's helpful. So maybe, I don't know how many people you can put on a Dropbox, or what's that other one that's free? Maybe some software program that we can all use, and we can put things on. I think we've both mentioned this to the NCI. Would they be willing to do some of this? And we haven't gotten answers yet. Because it would be wonderful if the NCI had a spot on their website and put all of this.
I think they're starting to do that within their CTSU thing. They have some collaborate sections where people can start different things, but I don't know who has who can start things, who can not start things, because working documents are on there. It's not very user-friendly, but it's a common place that all NCTN advocates should have access to, but I'm not sure. So
why don't we leave this kind of up in the air, but as you're thinking of things, please feel free to get the ideas to Patty and myself, because I think as a group now that we've come together every other week for a while, we're finding out that we can help each other, and that's what the purpose of these webinars are, is to tighten up what each of us knows and share ideas across each other, because each of us works in different places, and as we learn tips, you can bring them back to the table. And so anything that you can think of, please feel free to get back to us. But if we don't have any other questions, and at this point we don't, this is our usual disclaimer, and thank you always to Patty for help all the way through this process, and Jane for helping to